Okay, let's make a start. It's 4 o'clock, so we better start. So this is the continuation of the lecture that I started on Monday, and we were, to refresh your memory, we were going through the different steps in experimental design that we'd laid out. I think we've gotten up to about this slide. Anyway, let me go back one more slide just in case. Okay, but before we start, yes, I'm sorry I was a bit tardy in opening up the lectures on my learning, but they're open now for the first 3 weeks. We also have a lab next week on Tuesday. That's going to be on campus at 1 o'clock in 212. Okay. So I've opened up no, actually I haven't opened up the handout for that one. I will open up the handout for that one tomorrow, so you can take a look at that before the lab. Okay. It's just essentially looking at some of the toys that we can use to measure environmental, biotic and abiotic characteristics. Okay. All right, so for this afternoon, let's finish off this lecture. Finish off these steps in experimental design. The step number 7 is to obtain representative samples. Okay, so the key word being there, representative. We need to try and make sure that the samples that we collect when we make our observations are truly representative of the population that we wish to sample. Okay, and to make sure that it's truly representative, we need to be as objective as possible. Reduce the bias as much as possible. Reduce the bias in the way that we collect the data. All right, and quite often that means randomization of where we collect the data. All right, so when we randomize it or introduce some sort of randomization into where we collect the data, we don't have any biases going into choosing where we are collecting that data. All right, so randomization is a good way of creating a data set which truly represents what you're trying to sample. Okay, so randomization is really a good idea. Randomization can also help ensure independence of the samples. One of the key conditionalities of a lot of the statistics that we use, okay, is that the samples that we collect are truly independent from one another. Okay, they are not dependent. The position of where you take a sample is not dependent on where the previous sample or the next sample was or will be taken. Okay, you may think, well, I'd never do that anyway, but in some ways you can. So for instance, if you are sampling on a grid, okay, the first sample is really going to dictate where the next sample is taken, because you're taking it on a grid. You're taking a sample at each vertice on that grid, for instance, or the center of each square in a grid. So if you take a sample there, then we know the next sample is going to be in the center of the next square. So it's not truly independent, and the samples are not truly independent. Okay? So that means that the samples cannot be seen as really true representatives of whatever you're trying to measure. Okay? So we need to make sure that the samples that we take are independent of each other, and that particularly means that where we take them is independent of all the rest. The best way of doing that is to randomize the locations. Okay? Some ways in which we can reduce that randomization which is needed is by maybe doing stratified random. So that is an example where you have two different, say, habitats. You know that they're different, okay? But if you did a random sample, you would probably need to take more samples than you would necessarily need to adequately represent both habitats. The reason why you would take more samples than you need is because if you're truly taking random samples, then it's likely that you are going to take less samples in one type of habitat compared to the other, okay? And if you want to truly compare the two, then you will need to have the same number of samples. So you can imagine that it's going to be really difficult to get the same number of samples when you're sampling randomly, okay? So you would do a stratified random, where you take the same number of samples in each habitat, okay? But you would distribute those samples randomly just within that habitat. And in the other habitat, you would distribute the same number of samples randomly, okay? So that is known as stratified random sampling, okay? So in both cases, you have randomness, which ensures independence of the samples. The next thing you need to do is to obtain the data from those samples and then compare the data that you collected with what you would predict, okay? Right. We also need to think about experimental error. What you would need to do there is to imagine what sort of experimental error you may get in this methodology that you're using. To do that, you can take a look at the literature and see what experimental error was encountered in a similar sort of methodology, for instance. Okay? Experimental error may be natural experimental error, which is due to the inherent variability within the population, okay? Or it may be experimental error due to some sort of unconscious bias you've introduced into your measurements, maybe by using an incorrectly calibrated instrument, for instance, okay? You need to try and make sure that you account for all that experimental error, all right? You can do that by analyzing the data and taking a look at whether or not, say, the variability is too large to be accounted for this small or the large sample size that you take, okay? If the variability is too large, then chances are your sample size may actually just be too small to account for the natural variability in the data. So in that case, what you've got to do is to increase your sample size, okay? And you can do analyses to see whether that happens, okay? The second thing that you will do when you analyze the data is to compare your observations and the patterns in your observations to the patterns that could be predicted from your hypothesis. So remember, you have your research question, you formulate a hypothesis which can be tested, and you use that hypothesis to create predictions that your observations can be tested against. Okay? So your hypothesis says, okay, for instance, all the moths that we catch in this area will be black because soot covers the trees in this area and all the trees are black, so the moths are all going to be black. Okay? So the prediction then is all the moths in this area are black. All right? So the observations are, you go out there and you sample 10 different trees, well, the moths on 10 different trees, and you record their color. Okay? So at the end of it, you've got 24 different moths recorded, and each one of their color is black. Okay? So you compare that prediction, that observation to the prediction. To say you predict that all the moths are black, the observation says all the moths are black, so then you can say the hypothesis was not falsified, so the hypothesis is true. Okay? Now, that's just a simple example. Quite often, though, we use statistics. Okay? So we would maybe say that not all the moths maybe, how could I say that? Maybe move on to a different, yeah, let's move on to a different example then. That one's getting a bit difficult. We decide, we form a hypothesis that the size of the leaves of a particular species of bush is going to be bigger in a shaded situation where the light levels are lower, and they're going to be smaller in a sunny situation where there's more light. Okay? And the reason why we decide on that is because, well, we don't know for sure whether humidity plays a role or whether it's light level or so on. All we're testing is that leaves will be bigger in the shaded areas and smaller in the sunny areas. Okay? And those are the predictions. So we go out there and we measure two habitats, shaded conditions, sunny conditions. We would do a stratified random sample. We can use it straight away there. We measure randomly, position 10 points in the sun, 10 points in the shade. Okay? And we measure the leaves on a bush and get the, let's say, the average size of the leaf on 10 bushes in the sun and 10 bushes in the shade. Okay? Now, we can now compare the size of those leaves, all right? So we can use statistics to compare the size of the leaves. We can use a t-test, maybe, okay, or an ANOVA to compare the size of those leaves, okay? Now, if the average size of the leaf in the shaded area is actually bigger significantly than in the sunny area, okay, we have shown that our predictions are correct. Our hypothesis is not falsified. So our hypothesis that leaves are bigger in the shade is shown to be true, and we accept that, okay? Then we have to go on to why those leaves are bigger. Could be humidity, could be something else, okay? But we would use a statistical test then to compare what we find, our observations, with the predictions, okay? So even though the predictions didn't actually have any numbers, okay, we can compare the size of the leaves in the two habitats with each other and see whether that the prediction holds true. Okay? Does that make sense? Yeah. Okay. So the idea is then that we can use statistical tests. All right. So after we've done the statistical test, we need to interpret and draw conclusions from the data. All right. So we know that, for example, the leaves are different in size, but what does that mean? Okay. And in our discussion, we can talk about the different things that that means. Okay. From those discussions about what it could mean, we could then ask a next research question, formulate a next hypothesis, and do a next experiment. Okay, so if it was looking at leaves, size of leaves of the bush, okay, we see that they're different in light and dark. Now we can ask the question, what environmental factors in the light and in the dark are causing that difference in the leaf size? Is it light, or is it humidity, or is it the soil dries out quicker in the lighted areas? Okay? So we would need to formulate another hypothesis, which is testable, and form those predictions, and then get a methodology to collect samples or collect observations which we can compare to the predictions once again. Okay? So we interpret, draw conclusions. I think we test the scientific hypothesis back in 9, but I guess in this particular case, the idea is that we test the predictions, and then we formally say the hypothesis is accepted or rejected, okay? And then finally, we report the findings, all right? Now, I'm not going to gloss over that. Reporting the findings is actually very important, because if we don't write a paper on it, publish it, then nobody knows about it, okay? So reporting the findings and writing your research report is very important. Okay. All right, so we've talked about the different steps in the scientific process. I want to talk a little bit about designing the methods. Okay, so we've gone through going from the research question, proposing a hypothesis, which is falsifiable. You can see a way in which you can collect data to test that hypothesis. You form the predictions from that hypothesis, and then you now have to design a method to test those predictions or gather observations to compare to those predictions. Okay? This really ties into the lab next week, because I'll be asking you to say what sort of things you can measure from 5 different pieces of equipment, okay? With the idea of, okay, you can measure this with this piece of equipment. What predictions then can you test? Or what observations can you collect to compare to predictions? So therefore, what sort of hypothesis can you test with this piece of equipment? Okay? So there are many different types of data you can collect. You can collect sun, humidity, you can collect size of leaves, you can collect color of leaves, and so on. There are many different types of data you can collect. Okay? Which ones you want to collect, you need to be very selective about to answer the research question and test the hypothesis and compare to the predictions of that hypothesis. All right? So if your question is, like I was saying, the size of leaves, okay? Size, what sort of equipment do you think you would use to measure the size of a leaf? A ruler. Yeah, something like a ruler or a measuring tape, something which is calibrated to give you a standard length. Okay? So that would be the type of equipment that you could use. If you're looking at light level, on the other hand, what sort of equipment would you use? When in doubt, just put meter after whatever you're measuring. Okay? So in this case, meter. Or if it's humidity meter. Yeah, so you would use a light meter effectively. And that, I mean, the scientific name for that would be a light sensor, just give you a recording. It's a calibrated instrument. It gives you, it has a certain electronic output for the amount of light that it actually impacts on it. So it will give you a light level, okay? There are many different types of equipment to measure different things. Okay. I'm doing an experiment at the moment, and we are using a fluorescence meter. What that does is measures the amount of fluorescence that the photosystems in the chlorophyll of a leaf of a plant emit when you give it a standard amount or ultraviolet light, sorry. Okay, so from that you can work out what capacity that leaf has for photosynthesis, all right? So I'm testing to see whether these leaves, which are grown in the light, have a different capacity for photosynthesis compared to the ones, the same species which are grown in the light, okay? We find that maze of trees, their seedlings at least, grow better in the understory, in the shaded understory, compared to other species. It could be that they are adapted where chlorophyll, their photosystems within the chlorophyll are adapted to be able to capture more light, irrespective of the environment in which they're grown up in. So it's something which I'm learning about as I do it, but yeah, a question which I'm asking, okay? So there are different ways to compare observations to predictions. They're all mainly statistical tests, but you can also use things like graphical tests. So just simply comparing a graph, okay, which shows whether or not your observations are in one part, whereas your predictions are in another part of the graph, okay? So for instance, maybe all of the leaves that we've collected are large leaves, and we actually predicted that we should get small leaves. So in that case, we could say that just comparing the two graphs, that we don't really need to do a statistical test. We can see that all the leaves are different to what we expected. Okay, does that make sense? Yeah. So we don't actually need to do a statistical test. Okay? The thing that we're combating against, though, is this naughty thing, observer bias. Okay? We will display bias no matter what we do or no matter how much we try to get rid of it. Okay? Observer bias is always present. We will always try and limit it by doing things like taking random samples and so on, using calibrated objective instruments. Okay? But there may be some observer bias which sneaks in anyway. All right? So, for instance, if we're measuring leaves with a ruler, okay, they may think, oh, that will be fairly safe. But what if somebody decides, okay, I'm going to measure the length of the leaf from the 2 furthest points and on the leaf, so 2 different points. But another person approaches it differently and says, no, I'm going to measure the length of the leaf based on well, something else, which is not along the midrib of the leaf, but to the side of the midrib. Okay? So it's just a different method. Okay? But that is observer bias, because it could be that the different method of measuring the length of the leaf gives you a consistently different length of leaf. Okay? Or it could be that the person using the ruler measures not from the first graduation on the ruler, but from the end of the ruler. So all the observations are consistently longer by 5 millimeters or something like that. And the next person measures from the graduation to the graduation. Okay? So the lengths then are out. Okay? That's a type of observer bias. Okay? And we need to try and eliminate that. The different ways in which we can eliminate it by using these objective methods. Okay? All right. Effort and resources is also a very big issue with experimental design. The thing which constrains most experimental design is the amount of time and the amount of resources we have to collect the observations. Okay? Ideally, we would have an infinite number of observations. All right? If we had an infinite number of observations, then we could be 100% sure that we've collected we truly represented the area that we're trying to sample with no bias whatsoever. Okay? Unfortunately, that is not possible. We will always have to take a sample. Okay? And we need to try and make sure that that sample is has at least amount of bias as possible. Okay? Methodology using randomness is one way of doing that. Okay? However, I'm confusing myself and you with this now. I'm talking about numbers of samples rather than randomness. So the more samples that you take, the better. But the upper limit of samples is constrained by the amount of resources that we have. Okay? So the idea is to find that sweet spot, which is the minimum number of samples that we should take and adequately represent what we are measuring, the population that we are measuring. Okay? Any more, and we're wasting resources. Any less, and we're not adequately representing the population that we're measuring. So there are ways in which we can do this. And a lot of it has to do with the amount of natural variation in what we're measuring. Okay? If the variation between one part of the population and the other is quite high, in other words, there's a lot of variation in what we're measuring, then generally you need to take more samples, okay, to account for that variation. If the variation is, almost zero, so everything is pretty much the same, then you probably don't need to take very many samples at all. And if in fact you are 100% sure that there's no variation, then you could just take one sample and that would do it, okay? But of course you don't know how much variation there is. So what people would quite often do is do a pilot survey. So that a pilot survey is where you would take observations which or take a small number of observations, okay? From that, you could calculate what the variation is in the data. You have a better idea of what the variation is in the data. And from that, you can plug into formulas, which is quite often called a power analysis, which will tell you how many samples you need to take. Okay? So a power analysis is one way of doing it. You can do it also graphically by measuring things like, no, I don't have it here, a sample effort curve. Okay, sample effort curve basically looks at, sample effort curve, it basically looks at the amount of variation over an increasing amount of sampling that you do. If the variation falls and then levels off, and it doesn't change as you increase the amount of samples, then you've collected, you've got a large enough sample size. Okay? If, however, the variation is still continuing to fluctuate, okay, you still need to increase your sample size. Okay? So, minimizing the resources, very important. You need to collect the smallest number of samples to best or adequately represent the population. Okay? All right. Matching type of data to the predictions to be tested. Some of the things which you'll need to keep in mind is the objective. What is the objective? What are you trying to determine? The objective, I think, is can be thought of as what sort of predictions are you trying to test? Okay? Are you, for instance, determining the controls on distribution of an animal species? So what sort of data would you need to collect there? Okay? So the objective is controls on distribution of animal species. Okay, so the objective, so what sort of data would you need to collect? What sort of data do you think you would need to collect if you were determining the distribution of an animal species? Say it again. So maybe the other way around, you would need to understand how big an area you would need to house a population of these animals. Okay, so if you're looking at ants, for instance, you may get a population of ants within 20 square meters. Okay, so that would be the sample size that you need. Okay, and that may be the scale of your study. But if you are looking at trees, okay, and one tree occupies 20 square meters, okay, obviously a population of trees is going to be much bigger than the 20 square meters, okay? So you may find out that your size of your sample needs to be 5 kilometers by 5 kilometers, okay? You're measuring your tree, okay? It's actually a real problem when you are trying to research trees. You do need to have very large sample areas, particularly in places like rainforests where you have trees occurring at quite low densities, right? So you get one here, one 100 meters over there, one 200 meters over there, very low density. So to try and get some sort of sample size which catches the natural variation in the trees, you need a really big study area. Okay. Another way in which we can decide on the scale or the sample methodology is what sort of biological level of organization are we observing? Are we observing an individual, a community of that individual, a population of that individual, or are we measuring a community of different species of trees or birds or some other life form? Okay. Each means if we're measuring an individual, we just need to find one individual and measure that. Okay. But if we're measuring a population, we need to find at least whatever the natural variation is, maybe 10 individuals, maybe 30 individuals. Quite often, a rule of thumb is around 10 to 30 individuals may be enough. You could start there, okay? We're measuring a community. We may need to look at that community and decide what a minimum area is that we get all the different species of trees or birds or whatever within that community, okay? So whatever the methods are, it's going to influence how we collect that data. And really, what we're going to talk about in the next couple of lectures is how your methodology is going to change with the different type of data that you want to collect. Okay? All right. So pretty much done there. I've got a list of different questions on the remainder of this topic. I don't think I'm going to go through all of them, but I want you to go through all of them and have a go at answering them yourself, okay? If you want to show them to me and discuss them, that's fine, but just generally try and answer these things in your head, okay? So, for instance, how can I come up with a question? All right, we talked about that. Come up with a question by observations, just walking around, seeing things. You come up with a question by reading the literature and finding a gap in that literature, and so on. Okay? So another question could be, I have ideas and topics. but I don't have a project. Okay, so this might be a cry of a new graduate student. I have an idea, I have a topic, but I don't have a project. So I think what a grad student would have to do then is to come up with a research question, a hypothesis, predictions from that hypothesis, and then you would be able to set up your methodology to gather those observations to compare with your predictions. Okay? Now that would be a project. Okay? How can I refine a question so the answer can be obtained? And that's a really good question as well. And that is quite often an art. Okay? You get better at it the more experience you get. If you are just starting out, I would really suggest that you look at the literature. Okay? Try and find a question which is similar to yours and see how people approach it. Okay? How do they refine it? What sort of things do they look at? And then how do they match the methodology to that? Okay? Before too long, you'll become an expert and you will be able to refine the questions without referring to the literature. Okay? Next question, I have a project. Can I find an innovative way to look at the problem? So, in many cases, whether or not you're going to get your work published in a journal, which ultimately is how people judge your work, whether or not you have a paper and what sort of journal you get into, whether or not the journal will accept it, quite often has a lot to do with the methods that you use. If it's a novel method, an innovative method, then it's much more likely to be published in a prestigious journal. Okay? So that's why people like to use new methods to answer questions and problems and so on. Okay? The best way of doing that is to get up there to the cutting edge of science, as they call it, read the latest articles, and look at the methods which they used. Okay? And that way you would have a good idea of what the innovative methods are being used. Another way is to try and look at the observations which are being collected and see if there's any other way of collecting it. And you can do that by comparing what you want to collect with other disciplines. Okay? It could be that somebody in agriculture, for instance, has a different way of collecting data from the people who are collecting data in ecology. Okay? So if you could transfer that methodology to your topic area, it could be seen as a novel and innovative way of doing things. Okay? That's a lot of statistical methods have migrated from the social sciences to ecology and from ecology to the social sciences in that way. Okay? So a lot of these ANOVAs, I think, were first designed for the social sciences, and a lot of multivariate statistics like point-centered analysis, which you've probably never heard of, were developed for ecological analysis, and now they're used in things like economics and social sciences. Okay, so those are ways in which you can create an innovative way. How do I know whether I'm making a significant contribution to a body of knowledge? That's a good question. If you can look at the literature and not find the answer to the question that you want to ask, you can't find the answer to it, then you are contributing, you're making a significant contribution to a body of knowledge, okay? Even if it's repeating an experiment which has been done somewhere else here in Trinidad, it is still contributing to knowledge, okay? It'll be less likely to be able to get that published in a international top-tier journal, however, okay, because it's pretty much the same as what was done elsewhere, unless you get a completely different result with the same methodology that someone else got. And then that's interesting, because it means maybe that this area, Trinidad, is different to that area, okay? All right, how will I know whether my project is relevant, significant to a theory or a practical question, or whether your project is just stupid? Yeah, well, I think the best thing to do there is, if you're a new grad student just developing your projects, is to go and ask your supervisor. It's to ask the experts, okay? You can compare with methods used in the literature, okay? And you can ask experts as well, okay? Generally, if your methodology is going to give you observations, which allows you to compare to predictions, then it must be a correct methodology. And how can it be stupid? How can I conduct a literature review? I think whenever you write an essay or an undergraduate report or some lab report or something like that, you do a lit review, okay? So if there's one skill which you've learned at UWI, it's doing literature reviews, okay? And literature reviews are very important, as hopefully you get the idea of when we talk about how we know whether a research question is stupid or not, okay? How will I know what research is going on and what has been done? By doing a literature review. What about research in a foreign language? Interesting. Yeah, finding references in a foreign language can be very difficult. Yeah, I don't think I'm really going to talk about that too much. How will I know whether my project is too big or too small? All right, so if the observations that you need to collect necessitate you taking more samples than you have resources for, your project is too big, basically, okay? So if you need to take more samples than you have resources for, your project is too big. If you, on the other hand, can take the samples that you need and it's well within the resources that you have, then your project is a good size. Okay? How can I confine the question so that it is a master's topic and not a lifetime of study? And that is a very good question, okay? Ask the experts, I would say, okay? How will I know when to stop as an individual given the cyclical nature of science. So remember I was saying that, okay, we've found that leaves are bigger in darker environments and smaller in lighter environments. Okay. So the next step then, the next question you can ask is why? All right. And a testable hypothesis would be testing whether light makes the leaves bigger. Sorry, whether light makes the leaves smaller or not. Okay, and that could be using a greenhouse experiment, maybe something like that. Okay, but the problem is that you can keep on asking those questions. Why? 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 Eventually, you have to stop. Okay. When you decide when to stop is quite often a lot to do with how much time you have, okay, and how much time you have left. So if you are a grad student and you are happily conducting your experiments and you realize you've still got a year left in your postgraduate career, then there is probably time to go an extra step in the scientific process. Okay? Once you've done that, though, probably a good time to wrap up and stop. So you may get 2 or 3 papers out of that then, which is great. You've answered 2 or 3 hypotheses, tested 2 or 3 hypotheses, answered 2 or 3 research questions. You've gotten 2 or 3 research papers. Okay? Good time to stop. Quite often it is to do with the time limit, which ties into that last question there. How do I limit a project so it can be finished in 2 years? All right? Don't ask too many questions. Would be my advice to you then. All right, I'm going to leave it there. You can take a look through some of the other questions there. Okay. And thanks for coming. So next week, hopefully, we'll be able to get that tutorial moved. So we'll be doing it in the morning instead. Okay, but I'll keep you updated. Keep an eye on my learning. Okay.